Thank you, Munir. So continuing in the vein of rare diseases, I'd like to talk to you about pulmonary arterial hypertension, or PAH, as I'll probably refer to it throughout the talk. It's a rare condition and one with an unmet clinical need. The primary pathology is shown on this panel on the left. The arrow points to a precapillary pulmonary arterial, and you can see that the lumen is obliterated by cellular proliferation from the vessel wall. And you can imagine that this will increase the resistance of blood flow through the lung, and the impact this will have on the right ventricle, and patients die prematurely of right heart failure. There are drugs to treat PAH, and I'll tell you a little bit about those. These drugs, some of them are very expensive. Their use is regulated by 9 centres recognised for their expertise in the management of pulmonary hypertension in the UK. But you can see the challenge that these centres face when managing the disease. Their recent audit figures for December last year showed a 40% mortality rate at 3 years. So even the best centres, this is a challenging disease. You might ask why I'm interested in stratifying a rare disease, and the simple answer is because it shows clinical heterogeneity. So PAH can present as an isolated or relatively isolated entity when we refer to it as idiopathic PAH, or if there is a family history, heritable PAH, and that's shown on the yellow line here. It can present in the context of congenital heart disease, when interestingly it has a slightly better prognosis, and that's shown in the dark blue line. And it can present in the context of connective tissue disease of the lung and hepatic disease, when it has a somewhat more adverse prognosis. With drugs available, there is an interest in diagnosing PAH, and with that, the demographics of the disease has changed a little bit, and we are recognizing it in older people, and older people tend to have comorbidities, particularly hypercholesterolemia or insulin resistance, and that adds complexity to the disease and increases the mortality. And I mentioned that the product of pulmonary hypertension is right heart failure. When right heart failure develops, there are systemic consequences which also need to be taken into consideration when managing this disease. That said, we tend to treat all presentations of PAH with the same 4 classes of drug targeting the 3 major pathways shown here: the endothelin pathway, the cyclic GMP pathway, and the prostacyclin pathway, 3 pathways we've known about for the best part of 3 decades. We use these drugs alone or in combination. They do benefit the patient insofar as they relieve symptoms and increase functional capacity. And arguably, they may slow the course of the disease. But I don't think anyone's arguing that they arrest, let alone reverse, pulmonary arterial hypertension. And so there's considerable room for improvement here. There's one exception to that rather bland statement I made, or broad statement that I made, and that is we recognize a small number of patients with idiopathic PAH who respond to calcium antagonist therapy. We're talking less than 10% of that patient group, but they do show a marked reduction in pulmonary artery pressure in response to moderate to high doses of calcium antagonists, such as nifedipine and amlodipine. And they show an improved life expectancy, as you can see by the upper green line here. How do we recognize these people? Well, at the moment, we're dependent upon their acute response to a vasodilator challenge in the cath lab. In this study, they used inhaled nitric oxide, and you can see in the top 2 panels that those that showed a good response to nitric oxide, by which I mean a significant fall in pulmonary artery pressure or pulmonary vascular resistance, went on to respond to long-term calcium antagonist treatment, whereas those at the bottom that didn't respond did not respond to calcium antagonist therapy. We would like, of course, a molecular or biochemical signature of this responder group to avoid having to challenge them in this way in the cath lab, but that's not available at the moment. We don't understand the molecular basis of this responsiveness. That's not for want of trying, but we're just not there yet. Where we have made some progress is understanding the genetic architecture of PAH. And that began some time ago with family studies. And it's 20 years ago now since the publication of two seminal papers reporting mutations in BMPRZ, the gene encoding bone morphogenetic receptor type 2, as associated with pulmonary hypertension. So there's a family history of pulmonary hypertension in something like 6% of patients with PAH, and mutations in this gene have been identified in something like 80% of families tested to date. Since that time, family studies have shown mutations in at least two other genes. One is a transcription factor, TBX4, which is associated with small patella syndrome, and another is in the potassium channel, KCNK3. With progress in whole genome sequencing and access to this technology, we've been able to sequence a larger number of patients with pulmonary arterial hypertension. And in 2013, we began a cohort study funded by the British Heart Foundation, which, working with the Centres of Excellence for Managing Pulmonary Hypertension in the UK and 3 centres in Europe, we set about collecting very detailed clinical information about well-characterized patients, together with samples for a biobank for omic screening. So all patients were diagnosed according to international guidelines. And by 2018, we had over 1,000 patients in this cohort. You can see that around 6% had a family history, which is par for the course. Around the same number had a history of drug ingestion associated with pulmonary hypertension. And a small number of patients had pulmonary venous occlusive disease, which presents like pulmonary arterial hypertension, but doesn't respond very well at all to the drugs we normally use for PAH, and so are worth identifying. And I'll come back to that group in a second. But the assembly of this cohort coincided with the NIHR initiative, the Bridge study, which set out to whole genome sequence 10,000 genomes in patients with rare diseases. It was a precursor to the 100,000 Genome Project. And we were fortunate enough to have PAH recognized as one of the rare diseases in that venture. And that allowed us then to look very carefully at the genetic architecture of our patients compared to those others included in that study. And really, to cut to the chase, we were able to confirm mutations in BMPRZ, and not just in heritable, but also in so-called sporadic idiopathic pulmonary arterial hypertension. And in fact, mutations in that gene accounted for something like 15% of the patients in our cohort. So confirming this gene is perhaps the most commonly associated with PAH. We also identified mutations in other genes in the BMP signaling pathway, namely GDF2, which encodes BMP9, a ligand for the receptor, and SMAD8, one of the downstream signaling molecules. And so this, if you like, cements into our understanding of PAH the BMP-TGF signaling pathway, and an imbalance in TGF-BMP signaling is of major interest in the pathogenesis of PAH. But outside this pathway, we found mutations in another transcription factor in addition to TBX4. Those mutations were in SOX17, in ATP13A3, which is involved in polyamine transport, and in a water-poor aquaporin-1 in the cell membrane. And we also found a small number of patients have mutations in GCN2, which encodes a protein involved in the stress response. And this has been associated in family studies with PVOD. And so what we found by applying sequencing to our well-characterized PAH population, that we had misclassified, if you like, a small number of patients with GCN2 mutations who have a different disease, albeit presenting clinically like PAH. And the pathology in GCN2, EKIF2A4 disease is around the venous system rather than the arterial system. So this study has added a significant number of genes to our understanding of PAH. We now have something like 16 genes where pathogenic mutations have been associated with the condition. And so we've developed a picture whereby it clearly is an oligenic disease where perturbations in a number of pathways converge on a single endpoint clinical phenotype, PAH. And that's interesting because identifying these gene mutations opens up possibilities for new drug development to the extent that, of course, these targets are druggable. And that is a challenge. So we've known about mutations in BMPRZ for over 20 years, but it's only in recent years that we've managed to get close to developing some treatments which we might be able to take forward into clinical trials. There's a lot of discussion about how one might repair BMPRZ signaling. One suggestion is through repairing missense mutations through drugs like atilurin. We might be able to use molecular chaperones like phenylbutyrate or drugs which inhibit lysosomal degradation of the receptor, such as hydroxychloroquine. And there's interest in developing a ligand based on BMP9 for improving signaling through the BMPR2 pathway. I suspect some of these will, or I hope at least that some of these will come to early phase testing in the next couple of years. And in some cases, they may be very bespoke treatments indeed. So the work around the molecular chaperones and through inhibition of lysosomal inhibition might, or degradation, might be very bespoke to people with just a few people with a particular mutation in the BMPR2 gene. So we're watching this space with interest. But I'm interested in the sort of discussions that are going around that perhaps change our paradigm a little bit, or at least our view about this one genotype or one gene variant endophenotype, pathophenotype relationship. There are probably more genes or gene variants to be identified in our patient cohort, but we will need much more powerful technologies or much larger cohorts in order to be able to understand those gene variants, identify them, and understand their effect size. So I think attention is now turning to how one might use these gene variants with other omic data in networks that might better describe the patient populations that we're dealing with. And so what we're looking to is combining genetic variants with other omic data to perhaps pick out patients who might be vasoresponders as opposed to those that might have a more, for example, prothrombotic tendency. And so using network medicine, if you like, to describe in better detail the endophenotypes that singly or together contribute to the clinical presentation that we see and manage. And our first steps into this sort of venture have used proteomic data. So we were early adopters of the SomaLogic 1K aptamer program. And we used that technology to look at over 1,000 proteins in patients with pulmonary arterial hypertension compared to healthy controls. We were comparing levels in something like 250 patients with PAH. And not surprisingly, we found some differences which survived correction for multiple testing. And then what we've done with that is we've collaborated with colleagues at Harvard Medical School to map those differentially circulating proteins onto a protein-protein network. And the advantage of this is it allows us to map the proteins we've measured onto a protein network and identify connectors, if you like, other proteins which we might not have measured, which might also be part of a perturbed system. And then we can look for clusters. And one of the clusters that's fallen out of that is a complement cluster, which speaks to the complement alternative pathway. And if we look at how those clusters map onto our patient cohort, they appeared to identify 2 patient groups with different survival trajectories, lending, if you like, perhaps some biological context and biological importance to this cluster analysis. And this is interesting because the alternative pathway is the focus of attention from a number of pharma companies. We already have perhaps the most expensive drug on our formulary available to interfere with and inhibit the alternative pathway, but there are a number of others in development, and we're interested in taking this idea forward into our patient population. You can imagine how one could do a clinical trial relatively quickly. Somewhat encouraged by this, we've taken this network approach a little further, and again with our colleagues and collaborators at Harvard, we're looking at what we can do to create individual patient protein-protein networks, bespoke, if you like, to individual patients. And the concept here is that we take the proteome from a small number of healthy individuals to create a reference network, and then we map onto that or add on to that one patient at a time, their own proteome measurements, and to see how that then disturbs the network. And then we take those clusters out and map them onto a larger human interactome protein-protein network. This concept has been tested in the context of the transcriptome messenger RNAs, and we're eager to see how it behaves with our protein data, but that is a work in progress. I'll finish with just a couple of slides which show another value of mapping protein data onto the genome. And that is the Soma Logic platform is now, I think, expanded to 5,000 proteins. And a group at Cambridge have used that platform and mapped it onto to a gene sequence data set, and identified that for something like 1,400 of the proteins in that assay, there are genetic variants which have a pronounced influence on the levels of the circulating protein. And one of those proteins is of interest to us. It's a SNP which regulates the PDGF beta receptor. This protein, or this particular receptor, is of interest because it's a target for imatinib. And imatinib has been studied. It's been a phase 3 study of imatinib in pulmonary arterial hypertension, where there was an efficacy signal, but further development was halted because there were some concerns around safety with this drug in this patient group. And so what we've envisaged is that we might be able to improve the benefit to harm ratio by identifying a subgroup of patients that might respond to the drug, and we might be able to identify those using this SNP, which, as I say, has a pronounced effect on plasma levels of the target protein. And so the study we're just about to start is one where we recruit patients by genotype from our cohort. They've all been whole genome sequenced, so we can recall them by genotype, and then compare the effect of low-dose imatinib in the 2 genotypes to see if we can identify a biomarker for this drug. So I'll conclude there by saying that pulmonary arterial hypertension is a rare condition, but we're now thinking of it as a convergent phenotype. And our model at the moment is to try and understand how gene variants and the proteomic networks that we're creating describe these endophenotypes, which singly or collectively lead to a clinical presentation. The expectation is by understanding these endophenotypes in more detail, we might be able to come up with smarter drugs for this disease. Thank you very much.